At Easy Health, we talked about cellular respiration a lot regarding our product, but one key element to make all this work is photosynthesis. Photosynthesis can be separated into two parts: light-dependent reactions, known as light reactions, or light-independent reactions, also known as the Calvin cycle. Light reactions are directly associated with the absorption of light, while the light-independent reactions do not require light themselves. Light-dependent reactions start with a complex called photosystem 2. Photosystem 2 starts with a photon of light being absorbed by the antenna complex, which then transfers its energy to a P680 molecule, resulting in a charged P680 molecule. This excited molecule is transferred to the primary acceptor molecule, which is now negatively charged, resulting in a positively charged P680. The positively charged P680 molecule is extremely electronegative and can actually remove an electron from a molecule of water, resulting in oxygen being released and protons being released into the thylakoid lumen with a high proton concentration. This reduction of P680 is facilitated by the water splitting complex. The acceptor molecule then transfers an electron to plastoquinone and becomes neutral. The next part of the light reactions are known as linear electron transport. Plastoquinone shuttles electrons from photosystem 2 to the cytochrome complex, which uses its electrons to create a proton gradient. After the cytochrome complex, electrons are then shuttled to the mobile carrier plastocyanin, which also shuttles electrons from the cytochrome complex to its next complex, photosystem 1. Photosystem 1 is much like photosystem 2, where a photon of light strikes an antenna complex and is absorbed by a molecule, except the molecule is P700. When P700 is charged, it transfers its first electron to ferredoxin, which is an iron-sulfur protein. This oxidizes ferredoxin, resulting in the transfer of the electron to NADP+, reducing it to NADP. When the second electron is transferred to NADP by another molecule of ferredoxin, the second electron and a proton from the stroma are added to NADP by the NADP+ reductase to form NADPH. During the whole process just described, chemiosmotic synthesis of ATP occurs. What happens is protons are taken into the lumen by reductions and oxidations of plastoquinone. The splitting of water molecules adds 2 protons, and the removal of 1 proton from the stroma for each NADPH molecule formed decreases the concentration of protons in the stroma outside the thylakoid lumen. This results in a higher proton gradient in the lumen, resulting in a proton motive force that drives protons out of the lumen back into the stroma. To get outside the lumen, they must go through a protein complex ATP synthase. Where protons move through the ATP synthase, their free energy is captured and used to synthesize ATP from ADP and an inorganic phosphate. This process is known as chemiosmosis. Now, the second part of photosynthesis is the light-independent reactions, also known as the Calvin cycle. The purpose of the Calvin cycle is to produce glucose for the plants. The Calvin cycle happens in 3 phases. Phase 1, carbon fixation, phase 2, reduction, and phase 3, regeneration. Phase 1, carbon fixation, is when CO2 is converted from an inorganic to an organic form. It starts with an inorganic CO2 reacting with a molecule of ribulose 1,5-biphosphate, a 5-carbon sugar, to produce 2 3-carbon molecules of 3-phosphoglycerate. In phase 2, reduction, each of the 3-phosphoglycerate get an additional phosphate added on from the hydrolysis of ATP. This molecule is reduced by high energy electrons from NADPH, which were made from our light reactions, producing a glyceraldehyde G3-phosphate molecule, or a G3P. The final phase, regeneration, is where our G3P molecules are combined and rearranged to regenerate ribulose 1 biphosphate. So our cycle can repeat itself. The end goal of the Calvin cycle is to produce glucose. It requires 18 molecules of ATP, 12 molecules of NADPH to form a single glucose molecule from two G3P molecules.